Hello. The greatest single achievement of nature to date was surely the invention of the molecule DNA. And why does it make the greatest invention? Why is DNA the greatest invention of nature? Because DNA is the only molecule with inheritable information, meaning that DNA can undergo replication. Before going in there, we would try to have a philosophical touch onto what replication actually would mean. So in that, if you see replication here, I have for an experiment, for example, just for understanding's sake, we have a triangle, and somehow if this triangle has given rise to two triangles, is it replication? It must be. It has divided. It's something like a brick breaking into two pieces, formation of a new thing. That is replication in that sense. But then the problem is that they are not identical to the parent, or they cannot become identical to that of the parent. The same way, the difference in this scenario A, for example, is that one is a larger triangle and one is a smaller one. In other case, we have a triangle. I'll assume that they have same amount of matter, or they cover the same area. Assume that the area wise, matter wise, they are same, but the problem is that somehow they are in different shapes. So that means they are not again identical, or cannot become identical to that of the parent. In truest sense, what replication would mean is that it should be able to give rise to two daughter molecules or shapes or entities which look or resemble that of the parent. In organisms too, we can do that. A mother gives birth to a child, and the child certainly is not identical to that of the parent, at least in majority of the cases. The point here with reproduction in describing living organisms is that they have the potential to become as big and large as the parent. So I hope you got the idea that it should meet majority of the criteria to resemble, or the measures of resemblance with that of the parent. The progeny should look or have the potential to become that of the parent. In DNA, when we say replication and we say inheritable information, we mean to say that we have a DNA, say for example GATCAG, and the complementary of which would be CTAGTC. And if this molecule has given rise to two different molecules, GTCAAG, CAGTTG, and is this replication or not? In terms of number of bases, they are same. The composition of bases is also same. We have the same number of A's and G's in both the parent and the new molecule that has formed. The problem is that the sequence is not equal. The sequence is different, which would mean that they are not the same molecules. All of biological information is about sequences. The sequence of nucleotides in the DNA or in the RNA is the information. The sequence of amino acids in proteins is the information. The sequence will give rise to structural information. So in cases of DNA, sequence itself is the information, whereas in RNA or in proteins, we can say it as structures as information, but those structures are inherently dependent on the sequences. Of course, different sequences could give rise to similar structures. That is always a possibility. Here in replication, we should ensure that the two daughter molecules that form should share the same sequence information as well. I am Dr. BCMR, Assistant Professor at Shastra. We will discuss about the discovery of the replication. So as I said, the biggest or the most important discovery or invention by nature is replication. And DNA happens to be the molecule which can replicate. And that is how DNA is a good or fantastic molecule to study. We will try to learn the experiments that have allowed us to learn how this process of replication happens. There are three models that are proposed. Once the model was proposed by Watson and Crick, and structure is elucidated, and then the next thing came about how does the information of one molecule give rise to or is inherited by other molecules, or how are more molecules made with the same sequence. In the paper of Watson and Crick, they did describe about how the base pairing could influence everything. ATCG, if one of the strands has ATCG sequence, then it is the complementary strand would have a sequence that is determined by the other strand. This way, there is a possibility for inheritance of the sequence information. Nevertheless, there are several people who have proposed several methods, and these are some of the ones. One of it is called as the conservative method or model, and the other one is the dispersive model, and the other one is called as the semi-conservative model. So conservative model is the parent molecule will remain in terms of physical matter will remain as such, whereas the new molecule is made from new subcomponents or subunits or simply the nucleotides. In dispersive, the parent molecule is thought to have been broken into several pieces and then is shared by the daughter molecules, and the missing parts in the daughter molecules are made anew. In the semi-conservative one, it is proposed that the strands separate and each of the daughter molecules inherit one of these parent strands, and then the complementary of each of them is made. That is how two different daughter molecules are formed. So the now question is, there is to prove or disprove one or more of these methods, models, or see if all of them fail and there is something else that is still elusive. The experiments were carried out by Matthew Meselson and Meselson Stahl in about 1958. It's a paper published in PNAS. It's a fantastic work for the time where in those days people have to develop the technique, optimize, and then perform the experiments. Nowadays, we have too many techniques available at the will of the scientists, and they all have to do is just perform the assaying. And there is not so much of optimization as it used to be in those olden days. So for this is a very important, unique question. The DNA molecules are not invisible to the eye. And then somebody has to be able to say the process that is happening that is invisible. How do you do that? And that is what made these scientists great, and the experiment a very fantastic one. So they have chosen, we are going to discuss about some of the technical details, especially we would try to understand their experimental design, and I hope it will help us improve our own designing skills or research aptitude. The model organism they chose is E. coli, because by the time it was well established in the lab how to maintain, grow, and manipulate E. coli, and it has a very short generation time. It divides every 20 minutes provided the optimal conditions. And at that time they also found out they had available the N14 and N15 isotopes of nitrogen. The difference between N14 and N15 is N15 has an extra neutron. And then we have what they have is they also used a technique with ultracentrifugation, which would mean that they are going to use RPM of about or more than something like 40,000 RPM, and they would be doing it for several hours, sometimes about 16 hours or overnight. And there is one more aspect to it, is that they have used what is called as the density gradient. I hope the gradient is clear. And using cesium chloride, they were able to perform or make a gradient. What is density gradient here is, in different locations of the tube, there is a gradient of density within the tube, meaning that, say for example, this is 100% density. It would be somewhere in this location, it would be 80%, here some 60%, 40%, 20%, etc. Point is, the density and the bottom of the tube is very high, and at the top it is low. And this is going to go in the centrifugation, and it is going to rotate, and that is going to cause centrifugal force, because of which the molecules start moving. So in this experiment, the DNA was taken, DNA sample, and layered on top of the tubes, and then allowed to centrifuge. Say for example, if we had two different molecules here, the red is representing N15, and then the blue, or the say green one, is representing N14. N14 is light nitrogen, and we will call the other one, N15, as the heavy nitrogen. So what would happen? We will, just for the sake of understanding, we will just say that N15 is about 80% dense, and N14 is about 60% dense. And during centrifugation, when it is happening, they both will travel towards the opposite end, because now the tube is shown this way. It would have been in this direction, and it is getting rotated. During that time, say N14 will keep traveling, and once it reaches 60 density range of this in this gradient, it would stop moving there. It will stay there. Whereas N15, because it's heavier, because of the extra neutron, it would move till here, and it would stop there. So we are able to separate these two molecules. I hope you realize that it is not just the nitrogen N14 and N15 that are taken. DNA containing N14 or N15. DNA is a much larger molecule. It is a polymer of nucleotides. It's huge. And then if you have, and it has multiple atoms of nitrogen in the nitrogenous bases. So there are several millions of nitrogen atoms in DNA, and because of which the densities of DNA would change. N15 labeled one will have is highly more dense than that of the N14. So the experiment setup itself is like this, that they have grown E. coli in containing compounds that are made up of N15, because of which all the DNA and also probably the RNA and proteins also have nitrogen, so they also get labeled. But the point is that once we take out the sample, we eliminate all the RNA and proteins and other things. We only take the DNA. So although the other compounds or other molecules are labeled, it doesn't matter for the experiment. So they would have grown in N15 containing medium, and they would take a sample, and then they would transfer it into N14 containing medium. After that, they would take samples at different time points, something like 10 minutes, or they could take it 20 minutes or 40 minutes, and so on. So the samples are taken, and from each of these samples, DNA is collected, and that DNA is allowed to undergo centrifugation. And then they would want to see how many bands are forming. How thick are they? Are they light? Are they towards the light band or towards the heavy band? And we should also remember about the 20 minutes generation time. So here we can count it as 0 minutes, 0 generation time. Here it's about after 10 minutes, so we can say it about 0.5 generation time, and here it is after 1 generation, and here it is after 2 generations. So here let's have a quick look at about if there is DNA and if the 3 models were correct, assume dispersive, conservative and semi-conservative, what kind of banding pattern would we expect? If we just took DNA of that is grown only at N15 and only at N14, and we have taken the samples, it would give a band something like this, just for understanding's sake. The heavy one will be more dense, and this is where the axis of centrifugation and the molecules move that way. So N15, the DNA with N15 will be heavier than that of the N14 one. So let's try to draw what would happen after 1 generation, after 2nd generation, 1 generation, 2nd, and so on for each of the cases. Sorry. So in this case, after 1 generation, we have molecules containing DNA containing N14 as well as N15. So we would get a band somewhere here. This is where we can expect the band to be a diffuse band like this. What would happen in after 2nd generation? All the DNA molecules that we have would be sharing N14 as well as N15. So we'd only probably see more shift towards this side, towards the N14, because they are growing in N14, but we would see a diffuse band which is mostly moving towards N14. What would happen if it is conservative replication? In conservative replication, one of the molecules the molecule with N15 will remain same. The new ones forming are N14 type. After 1 generation, we should be able to see a band near N14 and N15 and 1 band with N14. And after 2nd generation, we would still see a faint band as such, and N14 band would be more relative to the band that is forming in N15. If it is semi-conservative, we will have a problem which would look, the band after 1 generation should be between N14 and that of N15, similar to that of dispersive. If they have stopped with 1 generation, then we wouldn't be able to figure out what would have happened. So what would happen in the 2nd generation is the question. What we will see is some bands are in the middle, and on some new bands have formed at the place where N14 band is there. That is what is expected. We can go on to the 3rd generation also and see what actually happens. So this is the whole experiment. You can also read it on Citable by Nature Education. It's a good resource for reading a lot of science. So here the experiment, as is already discussed, grown in N15 medium, heavy nitrogen, and then into the light nitrogen, that is N14. Then we collect samples at 0, at 20 minutes, and at 40 minutes. And then they have performed these experiments of ultracentrifugation, and they took pictures using UV light, and DNA would absorb at 260 nanometers. So you should be able to see a lot of absorbance at that place, and the other places reflecting it. Probably that's the photography they used at that time. And then a band that would have formed at N15 is here, that is at 0. At about 20 minutes, they found it to be in the middle between where N14 and N15 would have formed, and then there is another band that is after generation 2, that is after 40 minutes. What they found was 1 band in the middle and more on the light side, that is light nitrogen, which is indicative that which is conclusive that the replication is happening by a semi-conservative mode of replication. This is the actual figure that is published in the paper, and although it's not very easy for us to interpret it, it is nice to watch or look at the original data. We'll just try to see a little bit of it. So this is where N15 is, and this is the generation time, and you can see that some of the cells have already replicated and they're forming in the middle, 0.7 and this is one generation. This is N14 band, and this is somewhere, sorry, N15, and this is where N14 must have been there. So yes, light nitrogen. And after one generation we see a sharp band in between